So let's start a discussion of the energetic pathways, looking at the anaerobic pathways. This is the first pathway that's going to be used regardless of the duration or intensity. This will begin very quickly, almost instantaneous to the initiation of activity, and will continue for a short period of time. There are no organelles required for this pathway, and it is going to be utilizing a substrate level phosphorylation. The max time to exhaust within this pathway is somewhere between 120 and 180 seconds. We have two distinct types of pathways involved here. We have the phosphagen or the ATP-CP cycle, and then we have glycolysis and lactate metabolism. So we'll start here looking at the phosphagen ATP-CP cycle. This is the first energy pathway that's going to be utilized. It is completely anaerobic. It's limited by the concentration of ATP and the concentration of creatine phosphate within the cell. It takes place within and along the sarcolemma within the muscle and within the cytosol of the cells of other tissues. We will exhaust the CP cycle within about 6 seconds, typically due to enzymatic overuse and denaturing of the enzyme. So we're going to take a look here at the pathways, and then we'll talk about a secondary pathway that will come into play here. So we start off with the ATP-CP cycle, and we start with the hydrolysizing of ATP to free up phosphate, which provides the cell with energy to do whatever it needs to do. That phosphate is going to bind onto that CK enzyme that happens to be around, and the CK enzyme will link it to the creatine, forming creatine phosphate. The creatine phosphate will immediately lock back onto the CK, which will then strip the phosphate. As it strips the phosphate, the creatine dissociates, opening up a binding site for the ADP. We will then have the creatine kinase attach the ADP with the phosphate, regenerating ATP. Note the concentrations indicated for ATP, ADP, creatine phosphate, CP, and creatine. All of these levels are homeostatically regulated based off of the kilogram of tissue that we are referencing. The rate limiter for this pathway is the CK enzyme, not creatine phosphate. Creatine phosphate can act as a regulator of the CK enzyme. However, the CK enzyme is the actual limiter to the pathway. The pathway will reach exhaustion typically within 5 to 6 seconds. However, this pathway is trainable, meaning that when you have intensities that are beyond maximal, you can actually increase the efficiency of this pathway and get to about 8 seconds before the CK pathway itself reaches exhaustion. There is a myth out there that by supplementing with creatine, you're able to increase the rate that the system is utilized. That is actually not true. Because the creatine kinase CK is the rate limiter step, that is the true limiter to the trainability for the system, which means that you can supplement all you want, but unless you're training at high enough intensities, you're not actually going to get a benefit beyond what you can maximally get by yourself. Any study that has shown a positive effect typically has not taken into account the confounding variable of the training intensities that the person is working at. The high demand, we can go ahead and prolong this activity. We can prolong this activity by switching into what's referred to as the AK and AMPK pathways. So we have the AMPK and AK pathway. So once again, we start with ATP being hydrolyzed to allow us to go about doing some cell functions, leaving us with ADP. ADP can interact with a secondary enzyme AK, stripping a phosphate from another ADP, providing a phosphate to regenerate ATP, forming AMP. AMP can interact with an AK enzyme, stripping a phosphate, sticking that phosphate onto another AMP, allowing for the reformation of ADP, forming adenosine as the terminal product. This adenosine can interact with an AK holding a phosphate to form AMP, or it can be released from the cell. AMP has a secondary pathway that it can take beyond forming ADP. In this case here, it's where we have a free creatine molecule taking place. When the free creatine molecule interacts with AMP and AMP kinase, AMPK, stripping the phosphate from AMP, we get the molecule CP, creatine phosphate, formed. Adenosine will get formed as well, and that adenosine can once again interact with an AK to form AMP, or it can go about being released from the cell. CP can interact with CK, stripping the phosphate and sticking it onto the ADP to form ATP again, forming creatine and allowing for the pathway to start. This AMPK pathway is what takes place when you have very high intensity in the aerobic time intervals. The increasing levels of adenosine and the increasing utilization of AMPK is what leads to the feeling of fatigue and tiredness with increased levels of intensity. So let's take a look at where we're at. So we start with the APT cycle, and we cycle, and we're okay for a short duration. But what happens if we have to go beyond that short duration? This is where we're going to pull in the secondary anaerobic pathway, the glycolysis pathway, in which we're going to take glucose, we're going to pass it through a stepwise series of events to form pyruvate with the byproduct of 2 NADH H pluses and 2 ATP being formed. That pyruvate can go a few places, and we'll take a look at that when we look at glucose and pyruvate. Glycolysis. Glycolysis is a stepwise process. It is broken up into 2 distinct phases. We can look at this as a single pass where oxygen is available and pyruvate will go to Krebs, or we can look at this as a multiple path or lactate metabolism, where lactate is produced in order to regenerate NAD to allow for the continuation of glycolysis in an anaerobic state. If we look at glycolysis, we have 2 phases. We have the investment phase, where we are going to expend ATP in order to phosphate glucose. And then we have the energy harvesting phase, where we're going to reduce NAD to NADH, H+, and kinase ADP to form ATP. Note the end product of pyruvate and the byproduct of NADH and ATP. The overall biochemistry of this is an 11-step process. It's broken down into those 2 phases. The net result gain of ATP is 2 moles of ATP per mole of glucose. You can see the biochemical reaction laid out. And so let's take a look at this biochemical reaction. So time-wise, this is going to exhaust itself in about 30 seconds to about 2 minutes. Training and fitness level can take this up to about 180 seconds. The exhaustion is due to a change of pH, leading to changes of inhibition to key enzymes, a reduction of glucose intracellularly, an elevation of pyruvate intracellularly, or an elevation of ATP. The enzymes, PFK, phosphofructokinase, hexokinase, glucokinase, pyruvate kinase, and lactate dehydrogenase will be the enzymes that are rate limiters. The principal rate limiter is PFK. Glycolysis can be hormonally regulated by insulin shifting glycolysis towards glycogenesis, the producing of glycogen, or glucagon stimulating a glucose-sparing event. So let's take a look at the biochemistry of glycolysis. So we start with glucose, and from glucose we're going to form 2 moles of pyruvate. So notice I have labeled the first carbon and the 6th carbon in the glucose chain. So glucose comes in, interacts with hexokinase, stripping a phosphate from ATP and sticking it onto the 6th carbon, forming glucose 6-phosphate or G6P. P undergoes isomerization, forming fructose 6-phosphate. Fructose 6-phosphate interacts with PFK, stripping a phosphate from ATP, sticking it onto the first carbon, forming what's referred to as fructose 1,6-bisphosphate, or F16BP. This fructose 1,6-bisphosphate with fructose biphosphate aldolase forming dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. We've gone from having a 1,6 carbon sugar to now having 2 3-carbon sugars. We are now going to enter into the energy harvesting phase. But before we do that, we cannot use dihydroxyacetone phosphate in the pathway. We can only use glyceraldehyde 3-phosphate in the pathway, which means that we have to isomerize dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, which means we now have 2 molecules of glyceraldehyde 3-phosphate, and we continue the pathway in parallel once we are at this step. So the first thing we're going to do is we're going to stick a phosphate onto the molecule, forming 1,3-bisphosphoglycerate. In this process, we're going to be stripping away some hydrogens. Those hydrogens are going to reduce NAD, forming NADHH+. That NADHH+ will go to the electron transport chain. We then undergo our first kinase reaction, forming 3-phosphoglycerate, stripping the phosphate off of the first carbon. We have an isomerization reaction in which we go about moving the phosphate from the first to the second carbon, forming 2-phosphoglycerate. That phosphoglycerate undergoes a reaction. So we are stripping away water, forming phosphoenolpyruvate, or PEP. PEP then interacts with pyruvate kinase, stripping away the phosphate, kinasing ADP to form ATP, and producing the product pyruvate. So notice we started with 1 glucose and we ended up with 2 pyruvates. We have now terminated glycolysis. We have now taken glucose and metabolized it into pyruvate. So let's take a look at this pathway and let's figure out where we're going to see these rate limiters. The pyruvate itself has a couple of pathways it can go to, and we'll get to those pathways when we talk about pyruvate and glucose itself. The rate limiting steps are the first, third, and last step of the pathway. The rate limiting enzymes are hexokinase, phosphofructokinase, and pyruvate kinase. So let's put this all together in terms of the anaerobic pathway, including the continuation of glycolysis via lactate metabolism. So we start with the ATP-CP cycle. We then move into glycolysis forming pyruvate. Pyruvate will then interact with lactate dehydrogenase, forming lactate by oxidizing the NADHH+, reforming NAD. The NAD will then cycle back into glycolysis to allow for the pathway to continue. Now note that the lactate dehydrogenase pathway is a dynamic equilibrium pathway. Pyruvate will be forming lactate, but it will also be forming the acetyl-CoA at the same time. As pyruvate starts to accumulate, we start to produce more lactate so as to allow for the pyruvate to clear. If we get too much pyruvate, the pathway will slow down due to pyruvate inhibition on pyruvate kinase. So we're able to get ATP for about 180 seconds. And we're able to get ATP regeneration with high demand. Now, what's interesting here, regardless of what you may hear in textbooks, the lactate that is formed does not cause burning, nor does it cause fatigue. The lactate that is being formed will be exocytotic released, go into circulation, and will be utilized within the Cori cycle, and will be utilized at other tissues with lower demands for ATP that can reconvert lactate back to pyruvate and allow for aerobic metabolism to take place.